Let's try to sum up everything we have learned so far. First of all, glucose is converted into pyruvate by glycolysis. Then you have the decarboxylation step to produce acetyl coenzyme A that can join the Krebs cycle and eventually the electron transport chain for ATP production. But during starvation, you do not have glucose to undergo glycolysis. While in diabetes, you have similar problem because the glucose in the bloodstream cannot be imported into the cells. So the cells are starving and they cannot undergo glycolysis as well. But your cells will still require ATP to survive and to function. So these cells will try to switch on beta oxidation of fatty acids to produce acetyl coenzyme A, which can then supply some ATP production. But here is the problem. In starvation or in diabetes, your cells will try to produce new glucose molecules by gluconeogenesis. And since you do not have pyruvate, which is the starting material for gluconeogenesis, the cells will utilize the intermediate substrate from the Krebs cycle, which is oxaloacetate, to produce new glucose molecules. As a result, over time, your Krebs cycle is being drained by this process of gluconeogenesis. So in a normal situation, we can convert pyruvate into oxaloacetate by the enzyme pyruvate carboxylase. But that only happens when you have the surplus of glucose molecules. But now we're talking about starvation. So eventually, even the Krebs cycle is being shut off when we are running out of oxaloacetate. But your body is still producing a lot of acetyl coenzyme A from beta oxidation. Therefore, this acetyl coenzyme A must go somewhere. And we know that they are being converted into ketone bodies during starvation. So, ketone bodies can be used by your brain and liver during starvation or diabetes, but they are less efficient, meaning that they can produce less energy than through the normal oxidation pathway for acetyl coenzyme A. And they are mainly produced in your liver. So over here, you have the joining together of two acetyl coenzyme A forming a 4-carbon compound. Then you have another acetyl coenzyme A joining this compound forming a 6-carbon compound. But an acetyl coenzyme A is shortly removed to form back a 4-carbon compound, which is the acetoacetate that is unstable. So it will break into the 3-hydroxybutyrate molecule and an acetone. So collectively, these three molecules, acetoacetate, hydroxybutyrate, and acetone, are known as the ketone bodies. So one of the ketone bodies, acetone, is commonly found in the nail polish remover, and it has a sweet and fruity odor. And because acetone is very volatile, they can escape through your breath. So that happens commonly to the type 1 diabetic patients. So when the cells are starving, their body turn into ketone body formation. And that can be very dangerous because acetone is an organic solvent that can potentially dissolve away the plasma membrane. And in fact, ketosis has become the basis of the Atkins diet, which was first developed in the 1920s. So basically, in the Atkins diet, you have a low carbohydrate intake to trigger ketosis. With that, we force the body to turn on ketosis that burn the fat tissues. Okay, we have come to the final part of this lecture. Why do we get fat? Well, the answer is very simple. Instead of fat catabolism, we have fat anabolism. And the pathway for fatty acid synthesis is the reverse of the fatty acid degradation. So it begins with the activated acyl group and an activated malonyl group. So instead of cleavage, we have condensation that join these molecules together. Then instead of oxidation, we have reduction followed by dehydration instead of hydration, and again a reduction instead of oxidation. And finally, you will get the production of an activated acyl group. But before fatty acid synthesis can happen, we need to form the malonyl coenzyme A from the acetyl coenzyme A. So this is a committed step to start fatty acid synthesis. And it's done by the addition of a carbon dioxide in the form of a bicarbonate ion. So when you have both the activated fatty acid and the activated malonyl group, fatty acid synthesis can begin. But fatty acid synthesis is only switched on when you have the high levels of glucose, acetyl coenzyme A, and citrate, because these are the activator molecules for the enzyme acetyl coenzyme A carboxylase. And on a larger scale, the allosteric control are stimulated by insulin. So remember, you need insulin to increase glucose uptake by either the muscle cells or the adipose tissue, because it triggers the release of GLUT4 transporters. Conversely, when you get a lot of fatty acid production, such as palmitoyl coenzyme A, it can cause a negative feedback. And molecules that represent low level energy, such as AMP, glucagon, and adrenaline, can actually inhibit fatty acid synthesis. Because under this scenario, you would like to have energy production rather than fatty acid synthesis. In fatty acid synthesis, we use NADPH as the coenzyme, while in fatty acid breakdown or catabolism, we use NADH. That's because we want to have another set of enzymes and coenzymes for a better control of metabolism. So we are not expected to remember all these substrates and enzymes, but I do want to point out a common molecule here, which is the ACP. So ACP stands for the acyl carrier protein. Basically, it is a large protein molecule that binds the fatty acid chain as it is growing longer. So with ACP, it enhances the efficiency of lipid synthesis. Okay, here is a comparison table in between fatty acid synthesis and degradation. For synthesis, it happens in the cytoplasm, but for fat degradation, it is mostly in the matrix of the mitochondria. And in both cases, we need the activated compound. It is ACP, or the acyl carrier protein, for fat synthesis, but coenzyme A for fat degradation. Again, we use NADPH for fat synthesis, while for degradation we use NADH or NAD to be more specific. And in fat synthesis, we use the 2-carbon compound to form the 3-carbon malonyl coenzyme A. So remember, that is the committed step to start fatty acid synthesis. While in fat degradation, we have the production of the 2-carbon acetyl coenzyme A. With that, we have come to the end of this session. So to summarize, lipases break down triglycerides into its building blocks, which are glycerol and fatty acid. While glycerol can be used to make new glucose molecule by gluconeogenesis, while fatty acid can be broken down or be oxidized by beta oxidation, which is a 4-step reaction. And to build fatty acid, you have the reverse reaction, which is again a 4-step reaction. And the most important enzyme to remember in this pathway is the acetyl coenzyme A carboxylase, which is involved in the synthesis of fatty acid, and it is an allosteric enzyme that can be modified by several activator and inhibitor molecules.